Hello, everyone. Welcome back to Forensic Science, our little intro chemistry course to the basics of forensics. We are now in Chapter 13, looking into forensic toxicology. Glad to have you all here. As an introduction, forensic toxicologists are responsible for detecting and identifying the presence of drugs and poisons in the body, fluids, tissues, and organs. Forensic toxicologists not only work in crime laboratories and medical examiners' offices, but may also reach into hospitals, laboratories, and health facilities to identify a drug overdose or monitor the intake of drugs. Toxicology of alcohol. A major branch of forensic toxicology deals with the measurement of alcohol in the body for matters that pertain to violations of criminal law. The analysis of alcohol exemplifies the primary objectives of forensic toxicology, the detection and isolation of drugs in the body for the purpose of determining their influence on human behavior. Alcohol and ethyl alcohol is a colorless liquid normally diluted with water and consumed as a beverage. Well, alcohol can also be known as ethanol, used a lot in chemistry as a solvent. Alcohol is the most heavily abused legal drug in Western countries. About 10,900 traffic deaths each year are attributed to alcohol intoxication. Like any depressant, alcohol principally affects the central nervous system, particularly the brain. Blood alcohol concentration has been shown to be directly proportional to the concentration of alcohol in the brain. Alcohol's fate in the human body involves absorption, distribution, and elimination. Alcohol appears in the blood within minutes after it has been taken by mouth and slowly increases in concentration while it is being absorbed from the stomach and the small intestine into the bloodstream. When all the alcohol has been absorbed, a maximum alcohol level is reached in the blood and the post-absorption or elimination periods will begin. For an average human drinking on an empty stomach to modestly full stomach, alcohol is absorbed entirely into the bloodstream 30 to 90 minutes after the completion of drinking. When drinking on a full stomach, the absorption time can be as long as 2 to 3 hours. The type of beverage also affects absorption time. For example, beers absorb more slowly than 80 proof alcohol because of the carbohydrates in beer. The longer the time for complete absorption to occur, the lower the peak alcohol concentration in the blood. Alcohol distribution. When absorption is complete, alcohol becomes uniformly distributed between all the waterways of the body, which is about 2/3 of body volume. In decedents, if blood is not available, other watery portions of the body, like the brain, cerebrospinal fluid, or vitreous humor, can be used to determine alcohol levels. In alcohol elimination, this occurs throughout the body, is accomplished through oxidation and excretion. Oxidation takes place almost entirely in the liver, where the enzyme alcohol dehydrogenase converts alcohol to acetic acid and then into carbon dioxide and water. About 5% of the alcohol is excreted unchanged in the breath, urine, and perspiration. Ugh. Blood alcohol concentration is defined as percent weight per volume. Hence, a 0.1 or 1/10% is equivalent to 0.1 grams of alcohol per 100 milliliters of blood. That is an interesting combination of units. So gram per milliliter, sort of like density. All right, blood alcohol density. The elimination or burn-off rate of alcohol varies, but a 0.015% per hour seems to be a reasonable average. Hence, if your blood alcohol level of alcohol is 0.1%, takes about 6.5 hours for you to come down to zero. Blood alcohol concentration, BAC. The extent to which an individual may be under the influence of alcohol is usually determined by either measuring the quantity of alcohol present in the blood system or by measuring the alcohol content in the breath. The amount of alcohol exhaled in the breath is in direct proportion to the blood concentration and is directly proportional to alcohol in the brain. Blood alcohol concentration, again, the current legal measure of drunk driving in the United States is a blood alcohol concentration of 0.08% or 0.08 grams of alcohol per 100 milliliters of blood. Another way of expressing this is 80 milligrams per deciliter of blood. Not many people formally know the deci subunit. I think it goes liters, deciliters, centiliters, milliliters. All right. Alcohol of and the circulatory system. Humans have a closed circulatory system consisting of a heart, arteries, veins, and capillaries. Alcohol is absorbed from the stomach and small intestines in the bloodstream. Alcohol is carried to the liver, where the process of its destruction starts. Blood carrying alcohol moves to the heart and is pumped to the lungs. A simplified version of the human circulatory system. The dark vessels contain oxygenated blood, and the light vessels contain deoxygenated blood. Formally, what we have here is arteries and veins. What we have is, from the heart, we have the pulmonary artery take blood away from the heart to the lungs to pick up oxygen, and the pulmonary vein takes blood back to the heart. Therefore, our systemic artery can then pump oxygenated blood through the rest of the body, and then wherever there are capillaries where the blood is used and oxygen is absorbed and carbon dioxide is then picked up by the blood, it can travel through the systemic veins all the way back to the heart to restart the cycle. In the lungs, the respiratory system bridges with the circulatory system. A rapid exchange takes place between fresh air in the lungs, alveoli sacs, and spent air in the blood in the capillaries surrounding the sacs. If alcohol is present in the blood, it participates in the exchange, hence why alcohol can be smelled on the breath. As one exhales, carbon dioxide and alcohol leave the blood and oxygen enters the air sacs, known as alveoli. In the alveoli sacs, alcohol is in equilibrium with the blood. After leaving the lungs, the deoxygenated blood returns to the heart, where it is pumped to all parts of the body through the arteries, only to eventually return to the heart through blood veins, completing the circulatory process. Gas exchange in the lungs. Here this figure shows blood flows from the pulmonary artery into vessels that lie close to the walls of the alveoli sacs or alveolar sacs. Hence, the blood gives up its carbon dioxide and absorbs oxygen. The oxygenated blood leaves the lungs via the pulmonary vein and returns to the heart to be pumped throughout the rest of the body. They actually look like grapes. The respiratory system. The trachea connects the nose and mouth to the bronchial tubes. The bronchial tubes divide into numerous branches, primary, secondary, tertiary bronchi, that terminates in the alveoli sacs in the lungs. Breath testers. The temperature at which the breath leaves the mouth is normally 34 degrees. Breath testers operate on the fact that at 34 degrees, the ratio of alcohol in the blood to alcohol in the alveolar breath is approximately 2,100 to 1. In other words, 1 milliliter of blood contains nearly the same amount of alcohol as 2,100 milliliters of alveolar breath. Or 1 milliliter of blood contains the same amount of alcohol as 2.1 liters of breath from the lungs. How amazing. Breath testers that operate on the principle of infrared light absorption are becoming increasingly popular within the law enforcement community. Many types of breath testers are designed to analyze an individual's breath as it flows through a chamber in the breath tester and is exposed to infrared radiation or infrared light. It's the degree of the interaction of the IR light with alcohol in the breath chamber that allows the instrument to measure a blood alcohol concentration in breath. Some breath testing devices also use fuel cells, which converts alcohol to an electric current. Here we have an example of an infrared breath testing instrument, the Data Master DMT, quite an ambitious name. A subject blowing into the Data Master DMT breath tester. Schematic diagram of an infrared breath testing instrument. We have our light source for infrared radiation, or IR light. Breath is put into the inlet, into the sample chamber. Light passes through the sample. As we have breath going in out the outlet, as breath flows through the chamber, IR light passes through the breath and passes through a filter, then a detector on the other end, which gives a reading. The filter is used to, of all the different wavelengths of IR light released by the light bulb, the filter will select for wavelengths of the IR region, the infrared region, at which alcohol is known to absorb, and therefore we're able to find out how much light still makes it through and was not absorbed. Field testing. Law enforcement officers typically use field sobriety tests to estimate a motorist's degree of physical impairment by alcohol and whether or not an evidential test for alcohol is justified. The horizontal glaze, nystagmus test, walk and turn, and the one leg stand are all considered reliable and effective psychophysical tests. I would not do the reverse alphabetical because even I can't do that one with ease. A portable handheld roadside breath tester may be used to determine a preliminary breath alcohol content. These testers use fuel cell detectors and are not admissible in court proceedings as proof of intoxication. Fascinating. Now, testing blood for alcohol content, you can use gas chromatography, which offers the toxicologist the most widely used approach to determining alcohol levels in blood in forensic labs. Hospital and clinical labs normally use autoanalyzers to measure alcohol content. These instruments measure the conversion of alcohol to acetaldehyde by alcohol dehydrogenase enzymes. Collection and preservation of blood. Blood must always be drawn under medically accepted conditions by a qualified individual. A non-alcoholic disinfectant must be applied before the suspect's skin is penetrated with a sterile needle or lancet. Once blood is removed from an individual, the best way to preserve it is to seal it in an airtight container that contains an anticoagulant and a preservative, then store the sample in a refrigerator. Failure to properly preserve blood removed from living individuals may result in a decline of the blood alcohol level. In decedents, alcohol may be produced during decomposition, creating an elevated blood alcohol level. Alcohol and the law. All 50 states have established per se laws, meaning that any individual at a defined blood alcohol concentration, normally at 0.08%, shall be deemed to be intoxicated. At that level, a driver is 4 times more likely to become involved in an accident. At 0.15%, the chances rise to 25 times. To prevent a person's refusal to take a test for alcohol consumption, all states have adopted an implied consent law. The law states that the operation of a motor vehicle on a public highway must either consent to a test for alcohol intoxication if requested, or lose his or her license for some designated period of time. In Schmerber versus California in 1966, the Supreme Court ruled that taking blood samples was not protected by the Fifth Amendment. In a 2003 decision, the Supreme Court in Missouri versus McNeely ruled that police should obtain a search warrant prior to drawing a person's blood for alcohol testing. A diagram of increased driving risk in relation to blood alcohol concentration. Here we show that as the increase in blood alcohol concentration, even though just such a small percentage, can have an exponential increase in the likelihood for a severe crash or minor crash. Either loss of life, loss of organs and body damage, or loss of monetary gain if it's simply just crashing into a guardrail and causing multiple thousands of dollars of damage. So, drink very responsibly and have a friend to drive home if necessary. This is just other data how to tell what your blood alcohol level is after drinking. Basically, these linear line regions are taking into account one's weight and the ounces of 80 proof liquor consumed and the maximum alcohol concentration percent by weight, either on an empty stomach or a quasi full stomach. Role of the forensic toxicologist. Beyond the analysis of alcohol, the toxicologist is confronted with a maze of drugs and poisons. The toxicologist is presented with body fluids and or organs and is normally requested to examine them for the presence of drugs and poisons. Without supportive evidence such as the victim's symptoms, a post-mortem pathological examination, or an examination of the victim's personal effects, the toxicologist is forced to use general screening procedures with the hope of narrowing thousands of possibilities to one. Also, the toxicologist is not dealing with drugs at the concentration levels found in powders and pills, since anything consumed has been dissipated and distributed throughout the whole body. Furthermore, the body is an active chemistry laboratory, as few substances enter and completely leave the body in the same chemical state. Many drugs are chemically changed or are metabolized in the body to different chemical substances and at different rates of speed. Last, when and if the toxicologist has surmounted all of these obstacles, he or she must be prepared to assess the toxicity of the drug or poison. Because drugs constitute a large portion of the toxic materials found by toxicologists, much of the effort must be devoted to the extraction and detection. Many drugs fall into the categories of acids and bases. An acid, by one definition, is capable of donating a hydrogen ion or an acidic proton, and by some definitions, a base is capable of accepting a hydrogen ion or accepting an acidic proton. There are other definitions of acids and bases in the realms of chemistry, but these are the ones most commonly relevant to forensics. Acidity and basicity are both measured by the pH scale. By controlling the pH of a water solution into which blood, urine, or tissues are dissolved, the toxicologist can conveniently control the type of drug that is recovered. This approach gives the toxicologist a general technique for extracting and categorizing drugs. The analytical scheme, the strategy used for identifying abused drugs, entails a two-step approach: screening and confirmation. The screening step is normally employed to provide the analyst with quick insight into the likelihood that a specimen contains a drug substance. Positive results arising from a screening test are considered to be tentative at best and must be verified with a confirmation test. The most widely used screening tests are gas chromatography, GC, are an immunoassay. Gas chromatography, mass spectrometry are generally accepted as the confirmation test of choice. In gas chromatography, GC separates the sample into its components, while mass spectrometry, MS, represents a unique fingerprint pattern based off of the mass of each part of a molecule that comes off divided by the amount of charge on that molecule. To make a fingerprint pattern, fraction pattern, fragmentation pattern that can be used for identification, typically for a living person if it's the blood that is tested for drug content. However, urine drug testing for drugs is becoming common for jobs and employees in the workplace. Here we have the combination of a GCMS, which enables forensic toxicologists to separate the components of a drug mixture and provide specific identification of each separated component. So as we see in a gas chromatograph, on the gas chromatogram, we can see a mixture being separated into different components. And then once they pass through the mass spectrometer, we get different fingerprint filing or fragmentation patterns, which depend on the mass per charge that is developed. These can be correlated to what are typically seen for different elements, different molecules. They'll have their own fragmentation mass to charge patterns. Detecting drugs in hair. Hair is nourished by blood flowing through capillaries located close to the hair roots. Drugs present in blood diffuse through the capillary walls into the base of the hair and become permanently trapped in the hair's hardening protein structure. As the hair grows, the drug's location on the hair shaft becomes a historical marker for delineating drug intake. Human hair grows around 1 centimeter per month. Detecting non-drug poisons. Heavy metals such as arsenic, bismuth, antimony, mercury, and thallium may be encountered. Carbon monoxide is another common poison encountered. Carbon monoxide prevents the normal transport of oxygen throughout the body by sticking longer to blood cells than carbon dioxide does. In a healthy middle-aged individual, carbon monoxide blood saturation greater than 50 or 60% is often considered fatal. The significance of toxicological findings. Once the drug is extracted and identified, a toxicologist may be required to provide an opinion on the drug's effect and on individual natural performance or physical state. For many drugs, blood concentration levels are readily determined and can be used to estimate the pharmacological effects of the drug on the individual. The concentration of a drug in urine is a poor indicator of how extensively an individual's behavior or state is influenced by the drug. Before drawing conclusions about drug-induced behavior, the analyst must consider other factors including age, health, and tolerance. Often regarding a living person, the toxicologist has the added benefit of knowing what a police officer may have observed about an individual's behavior and motor skills. Drug Recognition Expert DRE program. During the 1970s, the Los Angeles Police Department developed clinical and psychophysical examinations that a trained police officer could use to identify and differentiate between types of drug impairment. This program has evolved into a national program to train police as drug recognition experts. Normally, a 3 to 5 month training period is required to certify an officer as a drug recognition expert. The DRE program incorporates standardized methods like blood pressure, pulse, and pupil size for examining suspects to determine whether they have taken one or more drugs. The DRE program, to ensure that each subject has been tested in a routine fashion, each DRE must complete a standard drug influence evaluation form. The DRE program usually cannot determine which specific drug was ingested. However, it is the production of reliable data from both the DRE and the forensic toxicologist that it is required to prove drug intoxication. Wow. So, just around the 25.5-minute mark, this concludes Chapter 13 of our Forensic Science College course. As always, subscriptions and likes are not necessary, but they are appreciated and more or less just funny to see. In any case, glad to have you all, and peace.